conditions and the stability of the vaccines is so important. Unless you properly stored and maintain the stability conditions, the expiry date of the vaccines or the degradation of the vaccines will be soon. Hence, storage conditions and the stability of the vaccines are so important in the preparation and during the storage. In this class or about the lecture, we will go into the how can you store the or what are the storage conditions of the vaccine and what are the stability, how can you increase the stability of the vaccines by adding certain agents. First, we will go about the stability of vaccine. The vaccines are always formulated so as to be both safe and immunogenic when injected to the humans. Whatever the vaccines we are preparing, it should be very, very safe and it should act as an immunogenic. That means it should act as an antigen so that the person can stimulate the production of antibodies. The vaccines are generally formulated as liquids, sometimes as a freeze-dried preparations, as in the form of the so that after the if it is a freeze-dried formulation, after reconstitution, and immediately we can prepare the solutions or about the suspension of the vaccine prior to the time of injection. And the vaccine is formulated with the bulk antigen. What vaccines we can add to the vaccines to increase the stability means we can add about the either the water or saline, the preservatives, and sometimes adjuvants, and sometimes we can also add the residual cell cultures. Okay, these are the agents which we can add, the preservatives, and sometimes we can add about the preservatives, adjuvants, and about the residual cell cultures. We can add these materials to increase about the stability of the vaccines. What are the preservatives we can use and what is their role means? Preservatives are used to prevent the contamination and to ensure the quality and potency of the vaccine during its shelf life. During its shelf life, okay, the preservatives, they will prevent the contaminations. As they prevent the contamination so that it can have the good potency and also the quality of the product. The second one is about the stabilizing agent, is about the adjuvants. The adjuvants help to boost the body's response to the vaccine. Okay, it's acting as an adjuvant. Next one is about the residual cell culture. These residual cell culture materials are used to grow the bacteria or the viruses to manufacture the vaccines. Okay, that is the role of the preservatives, adjuvants, and the residual cultures, cell cultures. These residual inactivating ingredients, they will kill the viruses or to inactivate the toxins, whereas the residual antibiotics prevent the contamination during the manufacturing processes. Okay, like this, these are the very important stability using the products to increase the stability of the vaccine. Whatever the new vaccines now are synthesizing, they are not containing any preservative. In addition to preservatives, some vaccines may also contain adjuvants. Okay, as we discussed, they enhance the immune effect of the vaccine antigen, but not themselves as an antigen. Whatever the adjuvants that we are using, they are not acting as an adjuvant. Just they are increases the immune effect of the vaccine. The examples of the adjuvants are about the aluminum salts are the most commonly acting as an adjuvant for the vaccine. On the other hand, we can also add the mercury, aluminum, formaldehyde, serum albumin, human serum albumin, and antibiotics, yeast proteins in the vaccines, which are not having any harmful effect on the instrumental or experimental animals also. Okay, here I have given certain examples of this vaccines, which is having certain the stabilizing agents or like the preservative, adjuvant, and the cellular residues, etc. I have taken the examples of the vaccines like the inactivated polio, hepatitis A, and hepatitis B. If you take an example of inactivated polio, the preservative is commonly used for these three type of vaccines is about the 2-phenoxyethanol. 2-phenoxyethanol is acting as a preservative for these three types of vaccines. And coming to the adjuvant, which is giving the booster or about the initiating the antigen, that is about the mostly we are using about the aluminum salts like the aluminum hydroxide or about the aluminum sulfate. This aluminum hydroxide is used in the hepatitis A vaccine, and aluminum hydroxide or aluminum sulfate is used in the hepatitis B vaccine as an adjuvant. Coming to the inactivating agents, certain vaccines they require some inactivating agents. For example, is about the formaldehyde. Formaldehyde is used as an inactivating agent in the vaccine known as the hepatitis A. Similarly, cellular residues, like the example is about the yeast protein, is used in the hepatitis B. Okay, these are certain stabilizing agents which can use in certain official preparations of the vaccines, which includes the preservatives, adjuvants, inactivating agents, and the cellular residues. This is regarding the stability of the vaccines. We will go or we will move to the storage conditions of the vaccines. Under the storage conditions, if you're not properly stored, okay, it causes the degradation of the vaccines or they lose their potency. Immunological products lose their potency on the storage if not properly stored. The preservation of the potency of immunological products, which involves the maintain the viability of the cells or preventing the denaturation of the protein, that is about the antigens or antibodies. If it is a vaccine, that is about the antigens, or about the seras, that is about the antibodies. Okay, we should prevent the denaturation of the protein and we should maintain the viability of the living cell. These are the 2 points are important under the storage conditions. The reduction in potency due to chemical changes which are directly proportional to the temperature of the storage. If the temperature or the temperature of the during the storage conditions, if not properly stored, there will be in the problem that reduction in the potency because of the chemical changes are taking place. Therefore, the temperature plays a very crucial role in the storage conditions. All immunological products are required to be stored below the room temperature. Okay, as we told that the chemical changes are taking place if properly not stored at the low temperature, there will be the problem is about reduction in the potency. Okay, therefore, all immunological products should be stored below the room temperature and should be stored in the refrigerator to ensure the reasonable life. Okay, we should freeze in the keep the or store in the refrigerator. The majority of the immunological products like the bacterial vaccines, toxoids, antitoxins should be stored in a hermetically sealed, light resistant glass containers. This is important. Okay, we should seal the vaccines in a hermetically light sealed, light resistant container which cannot be exposed to the light, dark colored, light resistant containers at the end should be stored at the temperatures between 2 degrees and 8 degrees Celsius. This is the most important temperature of which most of the immunological products are stored at this temperature. A few are the exceptional cases. The viral vaccines such as smallpox and oral polio vaccines are more stable at or below the freezing point. Okay, these are regarding the storage conditions of the vaccines.